back. So today I'm going to briefly introduce my story behind this imager work, which is also using an organoid to model the disease behavior. So the disease that I'm mostly focusing on about is prostate cancer, which is a highly prevalent cancer and the second cause of death for men with cancers in the US. And prostate cancer usually originated from the prostate glands, which can also invade it into other normal tissues. And in my research, I'm particularly interested in this phenomenon called cancer aneuploidy, which is featured by gains or loss of certain chromosomes in the cancer cell. And we assess the chromosome gains and losses in prostate cancer by using the data sets from Cancer Genome Atlas program. And as you can see here, some of the chromosomes have a higher levels of gain and losses frequencies than the other ones. So we always have the hypothesis that gains and losses of certain genes on these aneuploid chromosomes by changing their gene expression levels can promote prostate cancer. So to do this research, I've been trying several different 2D cell culture models. However, I'm facing a lot of difficulties and challenges. For example, if we grow, sorry.
I think this is what it's like to load some of these images that you're taking.
I see. Okay. So, okay, thank you. So, if we grow the prostate cancer cells on the dish, these cancer cells usually carry a lot of mutations and high levels of aneuploidy. And these mutations and aneuploidies can also evolve over time, which makes the studies very difficult and inaccurate. And if we grow the normal cells under the dish, these cells are usually grow very slowly, and then they will enter the senescence state, which is a state in which the cells stop dividing. And more importantly, the cells growing in the 2D cultures in this way will lack of many cellular signatures of prostate tissues. So to overcome these difficulties, I collaborated with Dr. Smith from the Vanderheiden lab from our Koch Institute and the Lee lab from the Brigham and Women's Hospital. And we isolated the primary prostate from desired mouse models and developed them into a multicellular organoids, which we can grow them as a many prostate organs in the cellular matrix, which we call the matrigel. As you can see here, by supplementing these organoid growth with a lot of growth factors essential for prostate tissues, they can grow pretty well in the matrigel, as shown here in the light microscope. And then in the stunning pictures that I'm showing, which didn't show up here for some reason, hold on. I use immunofluorescent staining and confocal microscopy and show that these organoids are actually actively growing by staining this Ki67 protein, which marks the cell cycle. And also these organoids expresses one of the very important prostate signature protein called estrogen receptor. And more importantly, I sequenced these organoids over time, and I found that they always showed a euploidy, which indicates these organoids are having a very stable and balanced genomes. So with these organoid models, I can introduce multiple genetic perturbations based on mutations and copy number changes in from the cancer cases. For example, here, I introduced the temporal fusion, which occupies 50% of the prostate cancer cases. And as you can see here, it causes some levels of morphological changes. And then we can do a lot of assessments to these organoids, such as evaluates growth and invasive behaviors and etc. And one of the most recent example is that, we just need to let it show up and over some time, is that we, should be fine. Okay. We identify some aneuploidy-associated gene. One of them is the RAD21 gene, but when gain of a copy can alleviate some of the cell death effects caused by the oncogene expressing or in the organoids, which in this way, it promotes the fitness and improve the fitness and the growth of the organoids. And then moreover, through a bridge project between MIT and Harvard School of Public Health, we are currently collaboratively investigating the hits that we identify in our organoid studies and see if they have prognostic predictions in the epidemiology studies for the patient cohorts with prostate cancers. And then we also identify some of the hits from the epidemiology study that we can bring it back to our organoid cellular studies to study the mechanistic aspects of these if this hits. So by these studies, we hope to identify some of the novel aneuploidy-associated markers in prostate cancers that may have some implications for therapeutics. So with that, I would like to thank my advisors, Drs. Angelica Among and Dr. Jackie Lees, and all my collaborators and all the institutes and funding resources, especially the Bridge Project, which sponsored my research. And thank you so much.